you. So I would like to break the monotony of the room. So whenever I go to conferences, I try to do the power pose. Sometimes like this.
Yes.
Because I just want to break the monotony of the room. And I'm not that serious, and you can laugh. That's fine.
You can dance.
You can dance. So I know that the hour is 3:30 CET and not the best time for a presentation, but I want it to be an interactive session for sure. And the topic that I have is design of experiments. Now I want to read the rule before I start. Who all have been involved in clinical trials directly or indirectly in the capacity of a doctor or any other capacity? Great, brilliant. So I think that I will make a little bit sense. So I am a biostatistician. I don't necessarily have a lot of knowledge about dermatology. But having said that, I have knowledge about data. And I have knowledge since last 13 years working on several clinical trials. And since last couple of years, especially on dermatology and cosmetology trials. So my objective today is to basically demonstrate how to methodologically plan a clinical trial in the way that the desired results that we want are attainable at the same time that it is also ethically approved by the regulators. So this presentation is going to explore several things that are key in any injectable clinical trial. I'm going to focus on injectable clinical trials because they have slightly different requirements than the normal drug products. So here we are going to talk about randomization strategies. We are going to talk about the control groups. We are going to talk about blinding techniques. We are going to talk about outcome measures. We are talking about patient selection criteria, regulatory endpoints, statistical design considerations. So to start, we are talking about randomization strategies. Now, why is it important? Anyone from the audience? Randomization strategies. Why not just take this? Let me take this room and this part of the aisle gets treatment A and this part gets treatment B. Why do we need to randomize? We need to randomize because we want to limit the bias. We don't want that the bias is introduced for the investigator or for the patient to know that which product he or she is getting. So there are different type of randomization that have also the advantage of balancing the confounding variables. So confounding variables are nothing but some variables that are like gender, your age, your Fitzpatrick test, etc. So the approaches could be simple randomization. It is as simple as tossing a coin. You could either get a heads or you could either get a tails. So each subject has an equal chance of assignment of treatment A or filler A and filler B. Similarly, we have block randomization. So when we are doing simple randomization in any sort of clinical trial, so we need a huge amount of data. The reason being that chance works when we have got enough data. Suppose, say, I am running an injectable clinical trial, which is fairly small in time, like 12 weeks, say. And by the 6th week, I want to see some sort of interim results. And this is a phase 1 or a phase 2 clinical trial, where we have a very small group. So here what happens is that if we are just going by chance, there is a high probability of finding the first 10 subjects to be having like 80% of them be having treatment A. So how to reduce that? We reduce it by applying some sort of blocks. So say in a group of 60 or 50 or 60 patients, we are creating some sort of blocks like block of 4 and block of 6 in which we maintain the randomization equally. So here we know that within a block, each and every participant is getting equal sort of treatment assignment. Next would be stratified randomization. Now, it has got a lot to do with the objective of the clinical trial. Now, suppose if I want to understand that how my filler is working or how my injectable treatment is working for people from the age group of 18 to 30, from 30 to 45 and 45 and beyond. This is my objective. So what should I do to achieve that? To have that, we add a layer to the randomization, which is called as stratified randomization, where we, along with randomizing per chance, we are also including a strata. Next is split-phase randomization, a very, very common technique in any sort of injectable clinical trial, in which one side of the phase gets treatment A or filler A, and the second side gets treatment B or filler B. So it has got, it is very common in clinical trials, as I said, and one treatment is on one side and another on another side. It kind of reduces the effort of having a lot of participants within the clinical trial while giving accurate results, because the same patient is giving two results. Thus, the confounding factors are kind of limited because the biology of the body remains the same. So next is control groups. So as we know that, without having any sort of comparison, without having any sort of control, it is very difficult to attain realistic or understandable results when we don't have anything to compare it with. So there are three types of controls. One is placebo control, another is an active comparator, and within subject control. So placebo control could be some sort of an inert substance like a saline prick, or it could be just a prick that is mimicking the treatment. Next could be an active comparator. That means that we are comparing two products. One is the investigational medicinal product, and the other is which is already available in the market. Then the next one is within subject control. As I said, it is the split phase design in which one side of the phase gets the derma filler, that is the investigational product, and the second side gets the one that is already approved and is in the market. Moving on. Now, blinding techniques. It is my favorite. I really enjoy speaking about this. The reason is that to reduce any sort of bias, it is very important to reduce the assessment expectation bias, and this thus to it is very critical in aesthetics where the outcomes are partly subjective. Now imagine that there in aesthetic clinical trial, it's not like your body that you cannot see your internal organ. You do not know whether your hemoglobin is getting better or not until unless you do not have your test results. In aesthetics, it is immediate. You get outcomes very quickly. Within a span of few days, you can see that which one is working and which one is not working. So here, I think blinding is very important. The reason, because both the investigator, the clinician, and the participant himself or herself can tell that which side is actually the IMP and which side is the placebo. So how can we do it? So the ones there are some strategies. One is the double blind, that is neither the participant nor the assessor knows about it. The next is evaluative blind, that is the independent trained raters assess photographic outcomes, as we saw in several of our presentations today. And the next is use of standardized photography that ensures consistent image capture across visits. Next is outcome measures. So without having any sort of outcome measure in our mind, that is the primary endpoint, right, for any sort of experiment that we are doing. First, we want to see that is there a scale that we would like to assess in this experiment to see what sort of grade improvement or what sort of efficacy or safety that we will achieve. So there are several in aesthetic clinical trials. As most of you are dermatologists, you must be aware about these validated scales that are there in the market. That is the wrinkle severity rating scale, the global aesthetic improvement scale, 3D skin imaging or elasticity tools or patient-reported outcomes. Next is patient selection criteria. Now, having a patient population where our results will actually work and demonstrate. Say I am developing a filler that would work for the nasolabial folds, but I'm injecting on people who do not have that. So it's not going to work, right? So having the patient selection in a way that we achieve what we are trying to show, to have the perfect inclusion-exclusion criteria depending upon our objective. When we think about inclusion-exclusion criteria in any sort of protocol, when I'm a non-physician, so whenever I'm planning a statistical analysis plan, I just run through it. But while I am designing a protocol, I have to understand that why this inclusion exclusion criteria is there. Say, if a person has already had some sort of procedure done on the nasolabial folds and is coming to the this sort of clinical trial as a participant, then it should be clear that whatever thing that we are going to inject, it may or may not have the desired results because there has been a prior treatment. So really formulating the inclusion exclusion criteria in any sort of protocol is very, very important. So some key factors for patient selection criteria could be age, gender, skin type. It could be baseline severity of any condition like wrinkle depth. So suppose if there are subjects, like we have got 50 subjects, out of which there are 10 with severe wrinkle severity, and we say that there has to be 2-grade or 3-grade improvement. It is very difficult because as you age, the wrinkles get deeper and the collagen gets low. So it is more difficult for such patients to achieve the desired results. So we also have to be realistic in setting our needs. Next is exclusion of prior aesthetic procedure in target area, because you may not get desired results or the outcome that you want, that is grade 1 improvement in wrinkle severity score. You're setting yourself for failure because you already had that at the beginning, right? You are not going to achieve that 1 grade because you're already coming with no or little folds. Next is allergy or hypersensitivity to the filler components. It's very, very important. Here is where our physicians and our chemists and all the other people in medical monitoring come into play. And they determine that what sort of components could be a component that could create some sort of allergies with comorbidities. So it's very, very important for patient selection criteria. Next is regulatory endpoints. So in regulatory endpoints that we are creating, they must satisfy the regulatory boards like FDA, EMA, and Health Canada. So there are already some guidelines for aesthetic clinical trials from FDA, from EMA, as well as from Health Canada. To align your clinical trial along with these guidelines is very, very important. Otherwise, there could be repercussions right from the first start, that is development of the protocol. So it is important to clearly, in an unambiguous way, to determine that what is your primary endpoint. So primary endpoint could be responder rate. It could be non-inferiority from a already standard product that is there in the market, or it could be superiority, depending upon what phase your clinical trial is in. Then the secondary endpoints could be onset time, duration of the effect, subject satisfaction, adverse events profile, and so on and so forth. So statistical design consideration. So here I would like to point out that once you start designing a clinical trial, it is very important to involve a statistician right in the beginning. No amount of statistical wizardry can rescue a clinical trial that has been planned in the wrong way. Because it starts from the inception, the statistical considerations like what should be the power of your study, what should be the power of your clinical trial. It starts at the inception. This is something that has to be decided on the table along with the statistician. So here, a robust design improves credibility and efficiency of a clinical trial for sure. So whatever we want to achieve, we want to have some sort of mathematical figure to it, right? When I say that I have lost some weight, it's ambiguous. I have to put a number to it. I have lost 3 kgs of it. Or if I say, oh, it's too hot, but how hot it is? Maybe it is 5 degrees Celsius more hotter than yesterday. So when we put a particular constant, it increases the credibility of a clinical trial. So these are some technical jargons and statistical mumbo-jumbos. But I will try to explain in a way that is, I think, that we do in the daily life. So sample size calculation. So say I just want to understand that what is the average age of this room. Okay. So or like of this entire conference. So I will take a sample of a couple of people and I will take the average age and I will determine what is the average age. So understanding that based on the endpoint and the variability and the desired effect, calculating the sample size properly is very, very important. What could be, I'm going to elaborate it in the other slides, that what could be the components when we are trying to calculate the sample size. Next is multiplicity adjustment. So there could be some cases where we want to have, like in a clinical trial, how many of you people have been a part of interim analysis, like a part of DMC, Data Monitoring Committee? Anyone? Have you seen any sort of interim analysis results? Like in between a clinical trial, interim analysis?
Yes, interim.
So in interim analysis, what happens that if you are, you are running a clinical trial and you want to see in between that you don't want to waste your time, you don't want to waste your money, you don't want to, put your subjects into some sort of danger to a prolonged time of that experimental drug exposure. So what we usually do, especially in phase 2 studies, is that we put a interim analysis where we want to see whether our drug is working, we can stop for futility. Or if we feel that we have got 2 or 3 drugs and if one is not working or the second one is clearly working, we can introduce that one instead of the other one. So what happens in such cases is that we have got an issue of multiplicity. Because when we are designing a clinical trial, we are putting a particular number that 5% chances are there that there could be an error. So when we say that alpha, when we say p-value alpha, I think all of us have heard these, jargons. But in simple language, we are saying 5% there is a chance of error. But for the entire clinical trial, right? But when we are stopping in between for at the interim, then we have to divide that, like 2.5% here, 2.5% there. So we have to also adjust that. Next is handling missing data. Now, I think that we had a lot of dermatologists today, a lot of doctors today, talking about follow-up, like having proper follow-up. As soon as people see that, okay, the results are wonderful, they stop, they won't come. So there is always, this is real life, this data that we collect in clinical trials is real life data. We see that as soon, like if the study is 12 weeks and by 8 weeks, everyone is having like beautiful results at their job and they won't come back from that. So, but our endpoint is analysis at the 12th week. So what should we do? Of course, we have to introduce something like missing data imputation. It could be as simple as like baseline carry forward, last observation carry forward, worst observation carry forward. We have to be sensitive to it. But it is always better when we are doing some sort of simulation of the data that we can do by some sort of MCMC model in statistics. I won't go into detail of it, but it kind of takes the average of every visit that the subject has, been for the derma filler and take the average rate of it as the endpoint. So I think here the investigators or the doctors, the clinicians, they should play a very, very crucial, they play a very crucial role that to inform the subject that how important it is for them to maintain a follow-up visit. Next is interim analysis. I think that I already covered that we include a data monitoring committee in which we don't want to put people at risk. We want to use the resources that we have in the most efficient manner. Next is statistics and clinical trial design are inseparable disciplines. So I think that statistics plays an important role, I think, everywhere. When we are talking about stock market, when we are talking about weather, whether we are talking about population in general, when we are talking about COVID cases, how many are rising, how many are falling, all these things have got like, some sort of statistics, it's numbers, right? So when we're designing a clinical trial, like I said, it starts at the inception. That what do we want to achieve, how we want to achieve, what are the clinical endpoints, how are we going to assess that? Now, say, okay, someone comes to me and says that we want to see that there's a lesion of some sort of skin, like there's a skin lesion. And I want to see that after applying my medicine, how much reduction of this, the lesion is happening. And I say, okay, very well, but how do you plan to record it? Because it is not just visible. When we are doing a clinical trial, we want valid data. And that valid data has to be dependent on some sort of validated score. It cannot be just something that we know visually we are seeing. Of course, AI plays a very, very big role, but it cannot do along with a already validated score that is already in the market, like an easy score, or you must be knowing there are several scores, right? So, statistics and clinical trial designs are inseparable disciplines. Each directly supports the success of other, but needless to say that designing of a good experiment comes first and statistical analysis comes second. So they support each other, but always the statistical analysis of good trial design. However effective a trial design is, essential and more important than just statistical analysis, a well-executed trial allows for accurate assessment of treatment effects, while a poor design leads to irreparable bias and imprecision. We know that in pharmaceutics, in cosmetic industry, so much of money is being pumped in creating these sort of treatments that we are trying to raise the bar of our safety and efficacy. But if we are not planning our clinical trial in the best effective way, what happens is that we are already setting ourselves for failure. I can give you an example from my real life. I was a trial statistician for a clinical trial in which they did not necessarily involve a statistician at every step of the way. Like CRF is case report form. I think all the doctors know that. They have got a questionnaire, they just record the age, BMI, things like that. And especially when it is like, some sort of clinical trial, then you have got even scales, right? So they did not plan for that CRF to be reviewed by a statistician. So the endpoint that they wanted to assess did not have it was not recorded within the CRF. And no matter how much they tried to manipulate, or I would say manipulate, because they were trying to manipulate the data so that they could take it out. A lot of medical monitors, a lot of people came in and they wanted to pick that data out from other patient reported outcomes and things like that, but they could not create that data that they wanted to use for the assessment. At the end, the study was discarded and stopped. So much amount of money and time, and this is an EMA study, and from a very well-known pharmaceutical giant. So here we have to understand that how and why these things are intertwined. So key advantages of a good design include meeting ethical standards. I think that it is the basic and no one can argue about it, because here we are actually working with patients. A little bit of irregularity in any sort of adverse events, any sort of patient profile will backfire. Next is efficient resource use. Needless to say, it is a lot of money that we are putting in, a lot of resources, time, and collaboration, and which must be used in a proper way. Next is isolating treatment effects. As the example that I gave you, that we want to isolate the treatment effects. Whatever is your objective, we need to understand that are we isolating the treatment effects? Like, I don't want to mix the wrinkle severity score with any sort of AE profile. That is two separate things. Efficiency is different and safety is different. Of course, we want both in the real world. Reducing bias and errors. Next is enhancing external validity. The aim is to provide precise estimate of treatment effect with a design that minimizes bias along with simplicity and credibility of the design and potential to impact future clinical practice. Now, needless to say, whatever we are doing, like in any sort of dermatology or cosmetology research, we want it to be reproducible. Of course, it is good for us, but it is also important as a regulatory, ethical standards. Because whatever data we are collecting, at any point of time, FDA, EMA, Health Canada can ask us to reproduce the same results on Earth. So there has to be no sort of, it needs to be unambiguous at all times. No confusion, clear standards. So enhanced design of experiments in injectable clinical trials. So can any one of you please, because you guys are from this, dermatology and cosmetology, can any one of you please expand on this, that why there's a need of enhanced design of experiments right now? Maybe it was not something that was seen 10 years back.
Because now patients, they want proper efficiency and no side effects, and there are so many companies which are, like I already mentioned, that off-label,
Yes.
And this is what happens then. If it is not designed properly or not statistically done, this is what happens. So of course it's very important. And of course, it's very important monophasic, biphasic, like, you must be knowing whether it is a deeper double filler or distribution or voluminizing, all that, yes, of course. So that when you categorize, it's easier for us to use in clinical practice.
Sure.
Yeah.
Anyone else from the audience?
There is a very regulation.
Yes.
There is the new regulation of medical devices.
Yes.
Which require the provider of the company to do clinical testing. In the past, it was not the case. I think it's also due to the.
You're absolutely right. Yeah. So, a few years back, if there was an open label study on a derma filler for few patients, it could get in the market. But now, with the increase in the surge of the new technologies, new molecules, new methods to do the non-invasive or minimally invasive, and uses of different needles, the size of needles, all these things really matter a lot in practice. And there's a very high competition in the manufacturers to attain that sort of FDA approval. This is a multi-million-dollar market of derma fillers, of injectables, and even the patients these days, they are very well informed about the ethical standards. They are very well informed about what is there in the market, what are the options. And when you want, when the manufacturers want to lay it out in front of you, they want to give you the best. They want to tell you that, okay, this is FDA approved, this is EMA approved, we had this results in a phase 3 trial. You go online and you look at the different publications of different companies who are doing these derma fillers. They are outright, stating the statistical significance that they found in 2 different groups when they did the derma filler clinical trial phase 3. So, and people like you, me, an average person, I think, of course, you guys are dermatologists, I am not. Even I go and I try to understand that, okay, what could be the side effects? I am a person who has got diabetes. I am a person who has got thyroid. Is it going to work for me? Is my comorbidity going to, intervene in between? That is why now designing an experiment in a way that we can optimize the trial quality, efficiency and reproducibility is very important. So we will speak a little bit of different types of designs of experiments. So the first one, as I said, is the split phase design. It is each patient acts as their own control, reduces variability, increases power. Ethical and efficient because fewer subjects are treated with the investigational medicinal product. Requires paired statistical analysis, of course. It is not an independent sample. It is not coming from 2 different people. It's coming from the same person. So we have to be considerate about the statistical analysis that we are doing. Next is the facial diagram showing us here the split phase treatment. Next is dose response and adaptive designs. Identifying the optimal dose and balance between the efficacy and safety. Now, I think people who are involved in clinical trials these days are very much aware about what is adaptive design. Who all have heard about adaptive designs? Okay. Do you know what is an adaptive design? Can you just share 1-2 lines with us?
It should be mentioned in the protocol in case, because sometimes companies do clinical testing and they notice during the protocol that they need to change. And if it was not mentioned before, they cannot be applied. And this is why I think FDA and European EMEA accept this new way of designing clinical studies.
Yes.
This is what I understood.
You're absolutely right. So, when we are talking about adaptive clinical trials, So we have to understand that, when we are doing some sort of injectable clinical trial, we are trying to work with some units, right? With some doses. That what is the unit that would work best if our wrinkle severity score is 3? What is the unit that will work best if our, aesthetically the scale that we are using is at the score of 4, that is like not that good. So here what happens when we are trying to adjust for the units and doses, we cannot we can't expose the subject to a particular dose, a high dose for a long period of time. It is not it, right? There could be so many, adverse events, as we discussed in the morning, that there could be some optical problems, there could be some nerve damage, there could be so many endless things, right? So here in adaptive clinical trial, what we are trying to do is that we start with 3 types of doses. Dose 1, dose 2, dose 3, and they are at different grades. How we are progressing is that we do an interim analysis and we see out of these 3 doses, which injectable is working the best. And then we say, out of the efficacy results, okay, the second one is doing the best. So we drop the first and the second. But at the same time, first and the third. And then at the same time, we have to do sample size recalculation, because we have dropped so many people out of the, the study. Either we put, apply the second dose to them, but then we will have to break it. We have to give them some sort of period where they are not taking anything, like, wash off period or some sort of, in between period. Or we can recruit more participants within the clinical trial. So ethically, it makes a lot of sense.
Which one is easier?
Easier is not adaptive design.
No, I mean, dropping those ones and getting new ones,
Dropping-
or just adjusting.
It depends, because what happens is that in aesthetic clinical trial, as such, getting samples, the participants enrolled is very, very difficult. So, I think that it depends. If you're getting participants in a certain site, I think dropping them would be better. And you can, have fresh participants enroll, because we never know, there could be some sort of, effect from the previous dose. And we don't want to confuse that. So, but as I said, that it is difficult to have participants within the injectable clinical trials, because even today, with so many advances in the injectable clinical trials, there is this type of, a little bit of hold back. There's a little bit of holding back of the participants to enroll themselves. Next, stratified randomization and patient selection. That is stratification by age, wrinkle severity, skin type for balance. It reduces confounding and improves precision, as we discussed previously. Patient eligibility criteria must define homogeneity, and clear inclusion-exclusion criteria improves reproducibility. So control arms in clinical trials. There can be 3 types, as we discussed. One could be a no treatment, placebo, or active treatment. Split phase can be product A and product B. Blinding techniques, as I said, that there could be an evaluator blind. So the assessors who are, assessing the kind of result that the injectable is going to have, they are not doing by in person. They are using some sort of anonymized photos of the participants, so that they can make a unbiased kind of assessment on. Next is patient blinding may be partial, so the dose on product masked. So patient blinding may be partial in the sense that there could be they may not know what it is at all, or they may know that okay, this is a vehicle, but they may not know that which type of vehicle it is. It is already a standard treatment within the market, but they might not know that which one it is. Next is using of identical syringes and masking labels. So I think that working in our clinical trial industry, we have easily seen that, placebo looks exactly, or the active treatment, the treatment B that we say, looks exactly the same as the treatment A. So there are techniques. I think packaging has got a lot to do, as we discussed, on presenting and, trying to mask it as much possible. Then objective tools like AI or 3D imaging also reduces bias. Outcome measures. So outcome measures, as we know, are clinician reported outcomes. that the clinician is actually using to tell whether there is an effectiveness or not. Then there are patient-reported outcomes, some sort of phase Q or satisfaction type of, CRFs that are filled by the participants. Then there is objective, which can be 3D imaging or elasticity meters, and then defining the responder thresholds, like greater than 1, greater than 2, what sort of improvement we are looking at. Innovations in the trial quality. So centralized blinded evaluation, I think that Irene did a fantastic job in explaining us like how the masking of the AI images, how helpful they are. Next is standardized injection techniques. I think it is very, very important. I think that doctors and innovators and researchers are working on that. Next is adaptive designs and interim checks for efficiency. I think, again, in injectable clinical trials, it becomes all the more important because here the timeline is very short and the outcome can be seen very, very easily, quite quickly. So we need to have that interim check in between to see which dose is working and which dose is not. Summary and takeaways is design of the experiment enhances the aesthetics, trial quality, and credibility. Thoughtful design is equal to less bias. Better endpoints, stronger approval pathways, use of split phase, dose modeling, objective tools, and standardized methods is the best way to go forward in both consumption of time and resources as well as getting ethical approvals. Now let's talk a little bit about the sample size calculation. Now what happens is, I hope I'm not boring you guys. Should I do the power pose again to entertain you? Okay, so let's talk about sample size determination, and I will not go into the detail about it. So when we are talking about sample size, we want to have a clear endpoint. We want to know that depending on the endpoint, what type of statistical test we can do that. It can be a two-sample test. It can be if it is a paired data, it can be a paired Coxon cyanide test. We want to know the effect. Then what is the effect that we are looking for? Is it like 2-grade improvement, 1-grade improvement, variability? We want to understand that in the previous trials, what is the variability that we had seen between the two groups? What was it like? What is the standard deviation? What is the deviation between the two groups? Then the alpha and the beta, that is the type 1 and the type 2 error, false positive, false negative. And understanding the dropout rate. Okay. As we said, that dropout is very, very common. So when we are doing sample size calculation, it is very important to consider 10%, 20%, depending upon the kind of clinical trial that we are doing. I had done once a clinical trial on Botox being used for spasticity. That is a neurological disease. You must be knowing it. So there was a company in US who were trying to use Botox to reduce the spasticity. They were using a lot of adaptive design clinical trial protocol. But the thing is that they had adjusted the dropout rate at 10. Now, when you are using a novel kind of thing like a Botox for spasticity, you have to understand that there will be a lot of dropout rates. They did not pay attention to it. It is a 2018 study, so it was long back. And now I think FDA is more and more stringent now. I think EMA, all regulatories are very stringent. As soon as they say sample size, they huddle together and they start talking robots about how to make the most efficient use of the people that are there. So why sample size planning is essential? So we don't want to overestimate our trial. We don't want to overestimate. Why should I put if I can do with 10 people, why should I inject the same thing into 30 people, right? And at the same time, I don't want it to be underpowered. I don't want 5 people to come for my clinical trial, and based on 5 people, I say, okay, I give a blanket on that, it's working, okay? By having a very small sample size, there are problems there are some adverse events that are rare. We miss out on it. Okay. So, there's a saying that a doctor is considered a good doctor not depending on the publications or the kind of medals he or she has, but upon the cases that the doctor has seen. So, I think that understanding if we have got a good enough, sample, we are able to understand what is the effectiveness what is the kind of adverse event profile that we can see in a new injectable clinical trial. So regulations require that the clinical or performance study plans include statistical justification for the sample size. Now, what is the justification for a sample size? So as I said in this previous slide, for having a sample size within the protocol for an injectable clinical trial, I have to go through all this. Now, is it something like a sample size paradox? Because here, what I'm trying to do is, for an injectable clinical trial, I'm trying to give you a sample, but the trial has not started. So how can I determine? It's a paradox, statistical paradox. So statisticians are expected to estimate sample sizes, though the trial has not started. What should we do in that case? There could be some treatments that are already there in the market. There could be some novel treatments. What should I do? So a biostatistician, what a biostatistician like me does is that goes to very similar products, very similar molecules, very similar therapeutic area, very similar indications, and studies the previous trials that have been conducted, understands the statistics, that is the average, the deviation that was derived, and constructs the sample size based on previous knowledge. Now, this itself takes a lot of effort from a statistician, especially in an injectable clinical trial these days, because all the previous ones were not necessarily done on regulatory guidelines. So, to design, like I will give you an example, there was a split phase sample size, split phase injectable clinical trial in which a split phase clinical trial in which the only population that I could get of reference was Chinese, and this study was going to be held in US. So how are you going to work? Here there's a very big deviation in the this thing. So it requires a lot of planning. Satish, am I out of time? Okay, I'll just quickly talk. Okay, the choosing the wrong sample size, either too small or too large, can severely impact the validity and the outcomes of the clinical trial. I think that we already discussed on the small sample size choices. So recruitment challenge in injectable clinical trials is that many acidic clinical trials target specific acidic needs. That is the nasolabial folds, lip augmentation, limiting the eligible participant pool. Okay. Next is ethical concerns. That is recruiting volunteers for cosmetic procedures. What happens is that generally the people who are volunteering are mainly healthy participants. They don't have any morbidities, right? But they are like enrolling themselves for this type of clinical trial. So FDA really requires to understand that the phase 1 of the clinical trial, that is the dose escalation, has proved that it is safe. Next is demographic imbalance. Studies often have a predominance of females, as we know, middle-aged and Caucasian participants, underrepresenting the age groups, the genders, and ethnicities. Next is placebo effect and blinding difficulty, because aesthetic challenges are visible, maintaining blinding and including a placebo arm can deter participants and complicate the study design. I will just move to the consequences and consideration. This is the last slide. So, underpowered due to inadequate sample size, the outcome and the conclusions can be compromised. Under-enrollment can cause clinical trials to miss significantly benefit or fail to conclusively demonstrate non-inferiority between products. Safety profile from small trials might appear clean simply because of a rare adverse event was not caught. So in summary, many global acidic clinical trials have faced enrollment shortfalls from phase 1 devices to post-market surveillance, that is phase 4 clinical trials, leading to early termination or limited statistical power. If you are designing a clinical trial in your capacity anywhere, I would welcome you to reach out to us, that is Symbio, and we would help you to design your clinical trial in the best possible manner. I think I can speak for myself that statistically I have got the experience, and my company is an end-to-end contract research organization. So thanks to Madness, and thanks to Symbio for having me here.